In this video at Medical Sciences Made Easy, we want to discuss about immunohistochemistry. This is going to be part 1 of immunohistochemistry, which is covered under histopathology. And this method is very sensitive and specific compared to hematoxylin and eosin stained smears. So I want to discuss in this session under immunohistochemistry. We are going to look at the principle of immunohistochemistry, which is normally abbreviated as IHC. We are going to see the principle, then we are going to see the procedures or the overview of the procedures seen under histochemistry. And as we begin, we remind you to subscribe if you haven't by hitting the subscription button, like the video, and share to others. And those who are interested in supporting this channel, you can join a membership by subscribing a certain amount every month. There is a provision of 10,000 and 20,000 if you want to support the good work so that we can continue to serve the community. So immunohistochemistry is broken down into 3 words. That is immuno from the word immunology. And immunology means antigen antibody reactions, or what we call antigen antibody complex. Whereas histo means tissue, comes from the word histology, which means tissue. Then chemistry comes from the word meaning it involves chemical reactions. This one involves chemical reactions by use of chemicals like dyes. We're going to see fluorescence dyes, enzymes, then different chemicals which are involved. That's why we call it chemistry. So that one comes from the word immunohistochemistry, which is the study of body tissues or proteins within a tissue. So we're going to see the tissue is the one which is going to be having the antigen, which are protein or tissue components, which we are going to direct a reagent containing antibodies. And when the reagent containing a specific antibody to this antigen epitope, they are going to react to form antigen-antibody complex, which is visualized by using chemical substances like fluorescent dyes, like enzymes, or radioisotope labels, or we can even use immunogold, or colloidal gold. So this is what we see the overview of immunohistochemistry. And after looking at this, now we are going to see the principle behind this. Whereby the principle, it states that this one is used, it is used to localize or identify or identification of antigen antibody reactions in a tissue which are visualized. These antigen antibody reactions in a tissue are visualized using labels, whereby labels, they can be dyes, fluorescent dyes, whereby here we shall use the fluorescent microscopy, or it can be by use of enzymes, or it can be use of radioisotope substances, and so on. So this is the principle behind immunohistochemistry, where we are seeing the identification or localization of antigen-antibody reactions. Localization of antigen-antibody reactions in a tissue. And after it is intubated to come up with these antigen-antibody reactions, these reactions are visualized or are seen using different chemicals, which we are mentioning here. After knowing the principle, now we must know the significance. Why immunohistochemistry? The significance of IHC. And the significance of this method, we are going to see the significance of this immunohistochemistry being, one of it is for diagnosis of cancers. We can use it for research purposes. We can use immunohistochemistry for differential diagnosis of cancers, whereby we can type cancers for differential diagnosis of cancers. We can also use it for prognosis, that is determining the progression on people who are on treatment to see if the drugs are working. Then we can also use to predict treatment, to predict success of treatment. So immunohistochemistry can be done to see whether if someone is given a certain anti-cancer drug or anti-tumor drug, there is a possibility of the person responding by using cancer markers, whereby we see if they are present and we localize them, then we know we can give a particular drug which will target and kill that tumor. Then lastly, we can use it for typing neoplasms. So these are the major applications of immunohistochemistry in medicine. After understanding the application, now we are going to look at the steps of immunohistochemistry. And among the steps we are going to look at tissue processing, or what we call tissue preparation. This one is going to be number 1. Then number 2, we are going to look at antigen retrieval. You can call it antigen retrieval, antigen, or you can call it epitope retrieval. From there we shall look at number 3, which is going to be blocking, or you can call it quenching. Then we shall look at number 4. Number 4 shall be antibody staining. After antibody staining, we shall see other steps like addition of labels. We shall see addition of labels or chemicals to visualize. Then after addition of labels, we shall counterstain with H&E, but this one is optional. Then after counterstaining, we shall look at mounting, whereby we dehydrate, clear, and mount. So these are the steps we are going to look at. These are the major steps we are going to discuss under the steps of immunohistochemistry. So without wasting time, let us begin with tissue preparation. And the specimen used in immunohistochemistry, we majorly use the specimen of choice is the frozen sections, whereby these ones are freezed at -80 degrees centigrade. That is how, in the presence of acetone, we use acetone. Then before using these frozen sections, it is always encouraged to bring it at negative 20 degrees for like 10 minutes, for at least 10 minutes to 20 before cutting sections. And sections are cut using cryostat. Cryostat is the microtome that we use for frozen sections. So in frozen sections, we use cryostat microtome for cutting sections between 6 to 8 micrometer. Then another sample we can use, we can also use formalin fixed, then paraffin embedded sections or tissues. Formalin fixed, paraffin embedded can also be used, whereby we are going to fix this sample using 10% neutral buffered formalin. We use 10% neutral buffered formalin, and after fixing it, we embed using paraffin. The tissue is embedded using paraffin, and it is sectioned using rotary microtome. So we have seen in frozen sections we use cryostat, but in formalin fixed paraffin embedded sections we can use rotary microtome or rocking or base sledge, depending on the type of the tissue. So this is the difference we can use. But the sample of choice is the frozen section because it preserves the antigens of within the tissue. Whereas the formalin fixed is not routinely used, but it is also used, but the best is frozen sections in immunohistochemistry. So this is what we can discuss under tissue preparation. And after we have prepared the tissue, we have come up with a section, and these sections are mounted on a slide using adhesive materials like albumin, like glycerol, or like cellulose. We can mount them. So after putting them on the slide, then we go to what we call antigen or epitope retrieval. And this process, which is number 2, is specifically for formalin fixed, paraffin embedded tissue sections. For frozen, it does not undergo into antigen or epitope retrieval. What is the principle behind antigen or epitope retrieval? It is that during fixation, we're going to see formalin will lead to what we call cross linkage, to bring about cross linkage and formation of methylene bridges. So this cross linkage and methylene bridges, they mask, these ones mask the epitopes of the antigen, or they cover. You can use the word they mask or cover the epitopes, so that during antibody staining, the antibodies cannot localize or cannot recognize the epitopes. So why we are doing antigen retrieval is to remove the cross-linkage and methylene bridges which are covering the epitopes, so that they are exposed for binding by the antigens. That is the significance of doing what we call epitope retrieval. And there are different methods used. Here the methods used, there are 2 common methods used, whereby we have heat-induced epitope retrieval, and there is what we call also proteolytic or enzymatic induced epitope retrieval. This is proteolytic. Here we are using proteases or enzymes. These are the 2 common methods used, whereby under heat induced we can use like microwave, we can use autoclave, we can use components like steamer and incubator. Majorly commonly we use water bath. Can use water bath And this one is done at temperatures of between 95 100 degrees centigrade. That is the temperature that is used. So we use at a temperature of 100 major degrees centigrade in a buffer. We use buffer also. So before heat-induced epitope retrieval, we first take section to water, then we bring it in this, and specifically it is done for 20 minutes. So that during heating, it is going to expose or to break this cross-linkage and methionine bridges to expose the epitopes. And after it has exposed the epitopes, it is ready for blocking. This is one of the methods, but it is not good compared to proteolytic. Why? Because antigenicity is lost, or the proteins within the tissue section can be lost due to heat. And another thing, it can lead to loss of counterstaining is lost. When you use heat-induced retrieval, there is loss of counterstaining, and then the loss of antigenicity can occur. That's why it is not good compared to proteolytic or enzymatic induced epitope retrieval, whereby here it is for endogenous substances. Like if we are blocking, we are going to use proteases like pepsin, we can use trypsin, we can use for blocking, for antigen retrieval, we are using pepsin, and there is proteinase K can be used. These are the major ones which are being used to retrieve the antigen. So these are the ones, and if these ones are done between, it takes 20 to 30 minutes, meaning it takes longer compared to heat, and it should be maintained at temperatures of 37 degrees temperature and pH, neutral pH of 7.4. So the condition temperature should be 37, here pH should be 7.4, which is neutral. But here we saw temperature of 100, here it is at 37 degrees, meaning heat-induced is very faster compared to proteolytic induced epitope retrieval. So this is the methods of epitope retrieval done on only formalin fixed paraffin embedded sections. For frozen sections, we do not undergo this step. Then the next step after looking at epitope retrieval, we go to blocking. Here we are blocking endogenous substances. We are looking at blocking endogenous substances in the tissue which might cause background staining. So here we are preventing background staining. So that's why we broke. That is the aim or the significance of blocking, to prevent background staining by quenching endogenous substances. And the endogenous substances which are blocked in this step include enzymes. We shall see enzymes like horseradish. We shall see enzymes like peroxidases. We shall see enzymes like alkaline phosphatase. They are blocked. We shall see non-specific proteins, endogenous non-specific proteins can also be blocked. We shall see blocking of biotin, endogenous biotin in the tissue. We can also prevent autofluorescence. So to block the peroxidase, in case the tissue has peroxidases, these ones are blocked. They are blocked using 0.3% hydrogen peroxide is added to block, and it can be for like 30 minutes. Then alkaline phosphatase is blocked by using, majorly we use levamisole or we can use 1% acetic acid. Here we can block alkaline phosphatase enzyme. Then non-specific proteins are blocked using 10% normal serum, obtained either from goat or sheep. This normal serum can block these non-specific proteins. Then another for blocking biotin, biotin is blocked by using avidin. So you'd stain using avidin to block the endogenous biotin. Then autofluorescence is prevented by using glycine or lysine, or you can decide to use non-fixative. You can use canoic fluid because this is a non-aldehyde fixative which can prevent autofluorescence caused by formalin, which due to porphyrin and flavins in the tissue. So here we can use canoic fluid to prevent autofluorescence. So these are multiple choice questions. You must master these, the endogenous substance and which chemical we use to block. These are very important to master as a student. So this is what we can talk about blocking, and the major aim is to prevent background staining. So after looking at this, now we are going to look at antibody staining. After we have retrieved, we have blocked the endogenous substances. Before we continue with these ones, let us talk about antibody staining. And under antibody staining, we have two methods, whereby method number one is direct method, or we can use indirect method. This is what is known as indirect IHC and direct IHC. Under direct method, here we are using only primary stain, primary antibody. Primary antibody is used only, and this primary antibody is always conjugated. It carries a label. Then under indirect method, here we use both primary and secondary antibody. So we first add primary antibody, we wash, then we add secondary antibody. So the primary antibody will be binding. If I can illustrate this, if this is a slide and it has a tissue containing antigen, so we shall come and add a conjugated primary antibody. And this conjugated primary antibody, then after certain, it will react to form antigen antibody complex, which is visualized. But under indirect, if this is a slide and we have an antigen here in a tissue, we are going to see we shall add first a primary unlabeled. This is a primary antibody which is unlabeled. Then later we add a secondary which is conjugated. So this one becomes secondary conjugated antibody. So these are the differences between direct and indirect immunohistochemistry. And we are saying, so this one becomes step number 4, which is going to lead us to step number 5, whereby we are going to expound on these labels added on these antibodies. So the 5th step is addition of labels or chromogens, depending on the type you have used. And we're going to see, in case you have used, in case you use enzyme as a label, and majorly enzymes used, we said it is horseradish peroxidase or alkaline phosphatase. These are the major enzymes. Why? But the most commonly used is HRP. Horseradish peroxidase is the most commonly used in immunohistochemistry. Why? Because it is small in size, very stable and easily obtainable. So the advantage of HRP is small in size, easily obtainable, and very stable. And when we use HRP as a label, we use the substrate which is added on this label. We add these ones are going to be called addition of substrate. That one, the substrate used for HRP, we use DAB, diaminobenzidine tetrachloride. We can call it 3,3-alpha diaminobenzidine tetrachloride. What we call abbreviated as DAB is the chromogen, the substrate chromogen, which brings about brown color. Brown color is formed. For alkaline phosphatase, we use nitroblue tetrazolium, which is abbreviated as NBT, and it produces the blue color where there is an antigen antibody complex. So this is part number one, when we use enzymes as a label, which are conjugated to the antibody. And these antibodies, which I have forgotten to tell you, that these antibodies can either be monoclonal or polyclonal. So the antibodies, how still have to put it, that they can be monoclonal or polyclonal. So we can either use monoclonal antibodies or polyclonal. Meaning monoclonal antibodies are made from a single clone of hybridoma cells. So for them, they target a single epitope. They bind only to a single antigen epitope. That is monoclonal. Whereas polyclonal, poly means many. So these antibodies, for them, they bind to different epitopes or different clones of epitopes. That's the difference between monoclonal antibodies and polyclonal. Monoclonal bind to a single epitope, whereas polyclonal binds to different or multiple epitopes. And we are going to see that the advantage of direct method is very easy. It is quick or faster compared to indirect, whereas it is less sensitive. So the most sensitive and specific is indirect method, is very sensitive. So as we continue with addition of labels, then in case we use fluorescent dyes, when we have used fluorescent dyes, and fluorescent dyes used majorly, we use FTC or rhodamine. And these ones, after applying them, we see the fluorescing substances using fluorescent microscopy. Here we use fluorescent microscopy for visualizing. Here we don't add substrate. Then in case we use radioisotope substances, here we shall use X-ray to visualize. And when we use colloidal gold as a label, colloidal gold is visualized using electron microscopy. Is visualized using electron microscopy. So this is what we can discuss under labels as step number 5. Step number 6, after we have labeled, we wash. And in each step, we wash using PBS, that is the phosphate buffered saline. For like 3 changes, we do 3 changes for 5 minutes. So after addition of the label, we can counterstain. And we said counterstaining is optional. This one, if desired, you do counterstain. And counterstain, in case you have used HRP, here you can counterstain using DAPI, counterstain. Then another stain which is commonly used is H&E. H&E stain can be used as a counter stain. Then you wash after counterstaining. Step number 6 will be dehydrating. You dehydrate. You clear using xylene, and mount. And here mounting, we can use DPX as a mountant. Dehydrating, we use increasing changes of alcohol. Here we are using increasing changes of alcohol. And this one marks the end. So after this, that's when we can visualize our outcomes of immunohistochemistry. So thank you so much for listening from the beginning until the end, where we have looked at what is immunohistochemistry. We have looked at the principle, which is to localize antigen antibody reactions or cell components or cancers by using labels to visualize majorly antigen antibody reactions. Then we have seen significance. Then we have looked at the steps whereby we have started with the tissue preparation, and we have said that the sample of choice is frozen sections, which are freezed at -80, and cryostat can be used. Then another one, we can use formalin fixed, paraffin embedded. Then we have gone ahead to look at antigen retrieval, which is to remove cross-linkage and methylene bridges for formalin fixed samples only. And the methods we can use heat-induced or proteolytic or enzymatic method, and we have seen different ways we can do it. And after this, we have seen blocking or quenching for endogenous substances, whereby we have shown you the substance and the chemical used. And we have said after doing this, we wash on phosphate buffered saline for at least 5 minutes in 3 changes. Then, and the major aim was to prevent background staining. Then we have seen antibody staining, which are of 2 methods, direct and indirect. And we have seen the advantage of this is quicker. This one takes time, but this one is more sensitive than this. And we have seen, we have even drawn to see how they look like. And we have seen different types of antibodies used, which we can use monoclonal or polyclonal antibodies. Monoclonal binding to a single epitope, polyclonal binding to different epitopes. Then we have looked at different labels and the compounds used. Then we have ended up by counterstaining and mounting. Thank you so much for choosing medical sciences to be your learning center. May the good Lord bless you.